And we continue with blood, right? So I hope up to this point you have watched all the prior videos. The last thing we covered was the blood clotting itself. So the thrombocytes and their intrinsic pathway for clotting, the fibrin mesh. I did upload as well two sort of supplemental videos, an animation that might give you a better insight on the process. And something that's near and dear to my heart, I work a lot with venomous creatures, is I uploaded a video, I put the link for the video that dealt with the effects of the venom on blood clotting itself. So again, the whole point of venom for a snake is to kill the prey. And well, how does it do that? What is the actual mechanism of the prey then dying? Well, check the video out, and I think you can apply then the knowledge that you know now with the thrombocytes onto the venom. So what do you think the venom stimulates? How does the venom achieve that goal of doing its effect there? So now we're going to move onwards to our sort of next bit. A real interesting part. I like this part of the chapter. We get into the idea of transfusions and blood replacement. So I suspect you have some idea of what this is. We're just going to get into some little details and also bring in the blood typing element to it. So a transfusion is defined as a transfer of blood or blood components from one individual, the donor, to another, the recipient. So this is, we're transferring blood or blood components. So normally, usually they involve the packed red cells. So when you do a transfusion, you're typically only giving the recipient the red cells, right? They remove some of the plasma and the serum, that kind of stuff. So let's say a person's at the hospital, they have an IV bag, they're getting the saline solution. That is not termed as a transfusion, that is an infusion. So introduction of fluids other than blood. So this is lactated Ringer solution. There's where they're getting nutrition, they're getting their ions, that kind of stuff. So different vocabulary means very different thing, the transfusion and the infusion. Right? So we're going to focus now our discussion on the transfusion. So if we're going to give blood from the donor to the recipient, there has to be compatibility. Right? So we're going to get into some compatibility rules here. So the patients, the recipients, can only receive blood that is compatible with their type of blood. So they can only receive blood that their body's not going to react against. So how do we define compatibility? Right? Compatibility is achieved if the red blood cells of the donor lack antigens for which the recipient would have antibodies against. So two new vocabulary terms. So antigens and antibodies. So antigen is going to be a foreign protein or a protein that triggers an immune response. The antibodies, which are going to come from our B lymphocytes, is going to go and attack this antigen. So kind of think of it, the antigen, the bad guy, the antibodies, the good guys, right? It's helping to destroy the invader there. Incompatibility occurs if the recipient has the antibodies against the antigen present in the red blood cells of the donor. Okay, so again, you cannot receive blood for which you have antibodies against. If you receive that blood, your body will mount an immune response, will destroy the blood cell that has been sort of transfused into the body. So if there is an improper transfusion, there's an improper cross-match, we have a condition called hemolysis, or some people call it hemolysis, right? Hemolysis is the rupture of the red blood cells. Again, that's not good. We don't want red blood cells to be rupturing in the body. And I hope you can kind of envision why, right? One, if you're rupturing red blood cells, you're not transporting oxygen, right? Two, if your red blood cells have been ruptured, what happens with all that hemoglobin, the iron, right? It's heavy. It's a heavy metal, and basically that will start to precipitate in the kidneys and the nephrons, and basically you're going to have now kidney problems as well, which can be fatal, right? So not a good thing. You do not want hemolysis. You don't want the red blood cells to be rupturing. So we must be very cautious when a cross match is done to ensure that we're transfusing the right compatible type of blood. Right? So again, transfusion reactions can occur when non-compatible blood is administered due to antibodies in the recipient's plasma against the antigens in the cell membranes on the donor's red blood cells. So if we have hemolysis, again, the red blood cells rupture or clot, and it leads to clogging of the small blood vessels, and the hemoglobin can again shut down the kidneys. So again, we have to be very careful in this cross-matching and transfusing process. So I have a little case study. I'll let you read that for a moment and see what you can kind of make sense of there. All right, so let's see what happened. So early in the morning, we have a 40-year-old male that presented. Seems like we solved the problem at 9:50. There was a cross match and a transfusion. All right, cool. We solved the problem. We're done. Yeah. Well, what happened later in that evening, right? 6:45. We're having chills, back pain, fever, and things go bad at 2:33. The person died. Right. So this is not the goal of an emergency room, right? To kill the patient. So what do you suspect was the cause of death, right? What do you suspect caused this 43-year-old man to die? So different factors, but I'm gonna suspect somebody made a fatal flaw in this case here, right? So the obvious sort of logical guess would be probably the cross match was not done properly, and the person was transfused with incompatible blood. So this is a simple little test, and again, had we been in normal situations, we would have selected one poor guinea pig or multiple poor guinea pigs there in your lab groups. We would have got a sharp lancelet. The poor volunteer, you would kind of then give us a sample of your blood, and we would subject it to the serum test, and we would then determine if you were type AB, type A, type B, or type O, right? And again, this is what the serum test would look like here. Right? So I'm going to come back to this slide. Let me give you some background information, and then this slide hopefully will make more sense. So when we look at human blood groups, most of you know about the ABO system. Some of you might even know your blood type. Well, I'm type A, I'm type B, type O, type AB. Right? So we know the ABO system, and a lot of you know the RH factor. So you're AB positive, you're type O negative, you're type A positive, type B negative, right? Well, there's actually more complexity to blood groups than what you might think, right? So blood is categorized into about 30 or more different blood groups. And these are categorized based on the antigens, the protein. I don't like the word agglutinogens. To me, that's a confusing term, agglutinogens. I like the term antigens, right? So the term antigens or proteins on the surface of the red blood cell. So again, the most clinical significant are the ABO, but there's other groups for which blood is frequently tested. These include the Lewis groups, the Lewis proteins, the Duffy proteins, the KID proteins, the KEL proteins. And this is not KID meaning a little child. The KID is the name of the scientist that discovered this. Yeah, but so these are, you know, they're important to be matched properly, but they're not as critical as the ABO. You can mess up on these and the patient will make it. They're going to have other complications, but they're going to survive, right? If you mess up on the ABO system, you're going to kill your patient. So you have to be very careful on the ABO blood typing, the cross matching, and we have to understand then the antigens as well. So let's see if this makes sense. So this bottom part right here, let me kind of address this slide here. So I don't know if you know a little bit about immunity. We're going to get to it in a later chapter, but right now in your system, you have antibodies for the tetanus bacteria. Right? How do I know? Because you couldn't have entered kindergarten without showing a vaccination record. Right? You may have never stepped on a rusty nail, but you got vaccinated for the tetanus bacteria. So normally we have to be either naturally exposed, you have to step on the rusty nail, or you have to get the vaccine. Somehow, some way, the tetanus bacteria must enter your body. And then we learn, we build memory, those antibodies are produced. So the prior exposure, right? That's the normal way things work. When we talk about blood, it works differently, right? So blood, for whatever reason, I don't have a good evolutionary reason for this for you, but for whatever reason, blood will spontaneously make antibodies, right? It doesn't have to be exposed. Maybe you've never in your whole life had a transfusion of type A blood. So you say, well, I've never been exposed to type A. If you're, just using the analogy, right? So if you've never been exposed to a different blood type, it would be logical to say you don't have antibodies for that blood type. But blood is weird, right? Blood generates spontaneous antibodies. Let me try to make sense of what that is. So let me start with type A, right? So that's the blood group A. So if you're blood group A, you are blood group A because on your red blood cell, you have the A antigen, right? So that's the antigen. We have antigens. We have proteins on our blood cells. So if you're type A, you're type A because you have the A antigen. Now, you've never been exposed to another type of blood, but for whatever reason, the blood plasma starts to manufacture anti-B. So we have anti-B antibodies that develop, right? Anti-B antibodies. So the antibodies, anti-B. So if you're type A, you develop anti-B antibodies spontaneously. What if you're type B? Right? You're type B because you have the B proteins, the B antigens, right? And if you're type B, you develop then the anti-A antibodies. What if you're type AB? So your body on a daily basis is used to seeing A, the A antigen, and B, the B antigen. So you're used to seeing A, you're used to seeing B, so you develop no antibodies for either of the two. If you're type O or type 0, right? O means blank, 0. You have 0, you have no antigens on your red blood cells. With no antigens on the red blood cells, your body's not used to seeing A, is not used to seeing B, so you develop anti-A and anti-B. Right? So the whole concept is weird here is the spontaneous antibodies, developing antibodies for which you've never been exposed to, for antigen that you've never been exposed to. So strange. So coming back here, so let's say that we subject serum. So if you're type A, right, and you subject anti-A, it's going to clot, and that's going to tell you, well, you're type A. You're going to subject type A blood to anti-B, it's not going to find anything, and it's not going to clump. It's not going to clot. When you see that little mess, that's showing you the clumping and the clotting factors. So if we subject anti-A to type O blood, nothing happens. If we subject anti-B to type O, nothing happens because there's no surface proteins on the red blood cells there. All right, so hope that's making sense. The idea of antigens and antibodies. So again, when we look at the ABO system, I hope you're okay with that. Then we look now at the RH factor, the rhesus factor, right? So rhesus factor was first discovered in the rhesus monkeys, and they then extrapolate the information over to humans. So the RH blood group is based on the presence of the C, D, or E antigens. So we have the A and B that make up the ABO system. The C, D, and E collectively make up the RH. Okay? So people with these antigens, if you have a C or a D or an E, you are considered RH positive. Right? You have one of these, RH factors, right? If you don't have a C, a D, or E, you are considered RH negative. So these are presence or absence. Present, yes, positive. Absent, no, negative. So unlike ABO blood groups, RH negative people do not automatically develop antibodies. This is a big deal, right? So we do not spontaneously develop antibodies. We must first be sensitized to those. So you must be exposed somehow to an RH factor that you don't have in order for you to develop the antibodies for it. So there must be some sort of prior sensitization or exposure before you develop antibodies. So very, very different category of antigens. So the ABO and then the CDE here. Okay, so let's look at some golden rules of blood compatibility. So recipients can only receive blood for which they don't have antibodies against. So you can only receive blood for blood which they don't have antibodies against. Recipients with an Rh negative blood can only receive Rh negative blood types. It's important, right? So one, you can have antibodies for the blood you're receiving. And two, if you're negative, you can only receive negative. Rh positive bloods can receive both Rh positive and negative as long as rule number 1 is followed. So again, the AB system is more critical than the Rh factor. If you're positive already, right? And I don't know, bad examples, but let's say you already have COVID, you're positive for COVID, and there's another positive COVID person over there. Well, you're already positive, they're already positive, let's hang out, I guess, yeah. If a person happens to be HIV positive, and there's another HIV positive person, well, we're already positive, we already got it, no worries, right? So if the person is already positive for the Rh, they can interact with the positive, and it's not fair to the negative, but they can also interact with the negative, right? So if I'm already positive for COVID, let's say, I can interact with other positive people, and it's not good for the negative people, but I could interact with a COVID negative person and it wouldn't affect me in any way. It would affect them, but not me. Again, bad analogies, I'm sorry for those, but that's what comes to mind right away. So again, golden rules. We can only receive blood for which we don't have antibodies, the spontaneous antibodies. And if you're Rh negative, you can only receive Rh negative blood. If you're positive, you can receive positive and negative. So let's see if this is making sense. So now is your little, sort of like a little quiz here, right? So give all the possible blood types you could administer if your patient, to your patient recipient is. So your patient is type A positive. Okay, so with as much detail as you can show, which blood types can you give to a type A positive patient? Right, I'll give you a few seconds there. So what blood types can be administered to a type A positive patient? Did you get it? Did you get it? Or did you kill your patient? I hope you didn't kill your patient. I hope you got all the types, right? Let's see. How many of you got those correct. Yeah, so these are the blood types that can be administered to an A positive patient, right? So the patient can receive type A blood. They can receive both positive and negative. Why can they receive the Rh positive or negative? Because they are positive, right? So if the patient is type A, you know that they're going to have anti-B in circulation. So they can receive nothing with the B antigen. So the A is good, positive and negative, and O, 0, has no B, so that one is perfectly fine to receive as well. All right, hope you did good with that one. If not, well, you killed that one, but let's see, hope you did better the next time, right? So now your patient is type AB positive. What blood type can be given to a type AB positive patient? Yes? Did you get it? Still thinking? Are you going to kill the patient? Hopefully not, right? Hopefully you're going to do good. AB positive is a kind of a famous blood type, and I don't know if you have AB positive. If you do, consider yourself very fortunate, right? AB positive is the universal recipient. Uh, if you're AB, that means you're not anti-A, you're not anti-B, you're anti-nothing, right? So you don't have any spontaneous antibodies. So type AB can receive type AB, can receive type A, can receive type B, and can receive type O. If they're positive, they can receive both the positive and the negative, the positive and the negative, the positive and the negative, positive and negative. So type AB positive, the universal recipient. So really, really good blood type to have if that's what you have. All right, next, O positive. What blood types can O positive receive? How'd you do? How'd you do? Right, it's O. So O would have anti-A and anti-B. So if you have both anti-A and anti-B, you know that you cannot receive any blood aside from the O blood type. So again, it's positive. So we have both the positive and the negative Rh that can be received. Now, if it had been O negative, the universal donor, right, they would only be able to receive the O negative blood. But if it's O positive, they can receive positive and negative. Right? So again, I hope this is making sense. I hope you're doing well. You're learning the golden rules of transfusions, and you're being able then to save your patients with the correct cross match and not giving them the wrong type of transfusions there. Right? Okay, so a slight different perspective. So we've been focusing on the recipient. Now let's look at the donor, right? So same concept, but just a slightly different perspective here. If the donor is B positive, which recipients can take his or her blood, right? So if you're B positive and you want to go donate blood, which patients can you give your blood to, right? So who can receive your B positive blood? Did you get it? Did you get it right? So again, if they are B positive, or I should say if you are B positive, people with B positive blood can receive your blood, but people with B negative cannot. Remember, a positive blood cannot be given to a negative recipient. So type AB, remember the universal recipient, if you're type AB blood, you can receive B positive blood. B positive blood can be given to AB positive patients, but not AB negative, right? Because again, the positive cannot be given to a negative blood type. If you notice O cannot because O has anti-B. A has anti-B. So a B positive blood cannot be given to an A blood type or a O blood type, only to B positive and AB positive. So again, I hope you see the little bit of a difference between these first three and now the perspective of the donor here. And last here, so the donor is AB negative. So if you're AB negative, who can you give your blood to? Right, so an AB negative blood can be given to did you get it? Yeah. So AB. So if you're AB, again, you cannot give to anyone that has anti-A, like a B type, or anti-B, like an A type, or an O, which has both anti-A, anti-B. So AB negative from the donor can only be given to AB negative and also to AB positive, right? So positive can get both the negative or the positive. So you're going to have more questions like this on your homework. And you're going to have questions like this on your blood exam. So again, be comfortable, be confident with these types of recipient cross-match, things like that. So again, hope that made sense. All right, so a few more slides to go. Almost done with this stuff here, but let's look now at a special case, a special scenario that we call the hemolytic disease of the newborn, right? Hemolytic disease of the newborn. That's a relatively new term. When I learned this way back when, they used to call it erythroblastosis fetalis. So I still have the habit of calling it that, right? Erythroblastosis refers to the red blood cells, fetalis to a little fetus, right, an unborn baby. When I think of blastosis, I think like things blasting, you know, so erythroblastosis fetalis or hemolytic disease. We know hemolysis, the rupturing of red blood cells, and this is happening in the newborn, right? HDN, hemolytic disease of the newborn. So going back to our rules, the compatibility rules, right? So mothers who are Rh negative, and the mom is Rh negative, she becomes pregnant. Her child, because of the genetic composition, maybe ended up being Rh positive. Okay, so mom has basically a foreign body growing in her her womb there, right? So her body has been Rh negative her whole life. Now all of a sudden there's this Rh positive. There's this CD or E protein developing inside her body. So towards the end of pregnancy, there may be a little bit of interaction between the sort of the fetal blood and the placental blood of mom. So mom may become sensitized to the CD or E antigens through childbirth. In childbirth, traumatic things are straining, and the placental may tear, that kind of stuff. So once the Rh positive blood interacts with mom's Rh negative, mom then becomes sensitized, right? So it becomes sensitized, and mom then starts to produce antibodies against that Rh positive. Now, this is a childbirth, right? So the first child, they're already gone, right? They're already out of the system, but mom has started to develop these antibodies against that. So first baby, not an issue. Let's say the second child is also Rh positive. So in this case, then mom, at a very early stage in the pregnancy, now starts to notice this foreign protein, which she already has antibodies against, and starts to attack this developing fetus, right? Causing the blood cells to rupture and clump, and it's not going to end well for that little fetus. So again, what is HDN? HDN occurs when a sensitized woman becomes pregnant with an Rh baby. So sensitized Rh negative woman becomes pregnant with an Rh positive baby. Again, kind of a picture here. So if there's kind of cross-contamination of the Rh positive into Rh negative blood of mom, mom starts to develop these green antibodies. First child is delivered, and then the subsequent pregnancy, the baby is at risk. If they're Rh positive, mom's antibodies will cross the placental barrier and start attacking the baby's blood. So again, not good. And in the olden days, I think a lot of women lost children during pregnancy because of something like this. So in the olden days, they would do blood tests to see if there was compatibility, that kind of stuff. Now they don't do that stuff because now there is basically a medication. It's kind of like an antibody that helps to desensitize the mom, right? So in case this event happens, the first child is okay, but now the antibodies that are produced by mom are distracted. They are kind of developed this antibody to prevent the second Rh positive child from getting attacked by mom's immune system. So it's a neat development of medication, and a neat type of immune response, knowledge, adaptation to prevent this HDN disease. So I'll post a little bit more detailed video. I'm a big fan of the Khan Academy videos. They kind of draw and give you a lot of detail on this, and it's interesting, right? If that pertains to you, ladies, again, it could make the difference between life and death of a fetus there, right? So, and with that, right, a lot of stuff to cover. I think we never really considered how complex blood was, right? There's a lot of things to cover with blood. We've kind of broken this up into several little lecture videos, but again, that should be enough to give you all the knowledge that you need to complete the blood homework and to do well on the blood section of the exam. Yep. Alrighty, so with that, I'll wish you a good evening, and I'll see you next time with the next discussion.